My name is Aaron Streese. I'm a professor at UC Berkeley, and my lab develops technology to study biology at the single cell level. Why do we want to measure single cells? Well, high throughput measurements, precision measurements on single cells and technology that can make those measurements are critical for large-scale research efforts such as the Human Cell Atlas Project, which is one of the primary research directions of the Chan Zuckerberg Biohub. And the goal of the Human Cell Atlas Project is to characterize or map the cellular composition of not only healthy humans, but also to be able to measure the change in cellular composition across human genetic variation and in response to perturbations such as disease, drug treatment, and aging. And tools that can make high throughput measurements on single cells for such large scale efforts need three things. They need to be high throughput, be able to measure many cells at the same time. They need to be able to be sensitive, to make measurements on very small amounts of material, and they need to be precise, be able to make the same measurement over and over and over again. And so the molecule of choice for characterizing cell state has been RNA, and technologies for high throughput single cell RNA sequencing have really pushed in the direction of these two axes, really throughput and sensitivity, the ability to be able to make more measurements on more molecules. And if you look at the progression of throughput in this space over the past 10 years, the inflection point in the number of cells, here I'm plotting the number of cells that can be measured in an experiment, the inflection point occurred at the implementation of microfluidic technology, or microdevices. This is a suite of technologies that sort of ride on the back of the silicon manufacturing industry. And instead of making integrated circuits, we can make microdevices for high throughput biotechnology applications. And so my lab has been focused on pushing in these two directions, increasing the throughput and sensitivity of single cell sequencing. But recently, we've also been interested in trying to develop technology along this third axis, which is being able to make more measurements on different kinds of molecules in the same cell in a high throughput fashion, with the goal of being able to characterize molecules that give us information about the epigenetic state of cells and then also the output of genome regulation to measure protein composition and the composition of metabolites. And we do that by integrating nonlinear optical imaging techniques with microfluidic platforms and genomics so that we can make these multimodal measurements on single cells. And when it works, we also develop algorithms for analyzing these large, highly dimensional data sets. And when that works, we look for collaborators so that we can apply our technologies to study interesting tissues like adipose tissues in the immune system. So if you're interested in working with us, please look me up after the session. Thank you very much.